Hello student, welcome in the next lecture of our discussion where we are discussing about plant adaptations, and we have started our topic about hydrophytes. So we have discussed about the morphological adaptations, and we have started discussing about the anatomical adaptation of submerged hydrophytes. Now in this lecture we will discuss about the floating hydrophytes whereas amphibious hydrophytes. Okay. So coming to free floating hydrophytes. Now free floating hydrophytes, definitely they are floating on the surface. Okay, their leaves, basically their leaves are floating on the surface, and they have roots which are inside water. They could also be that case where the leaves are there and they are attached to the surface of the water body by roots. So that both conditions could be there. So we will try to analyze the anatomical structure of both the conditions. Now in this floating hydrophyte, first of all, roots. See, in this case also root will be poorly developed because if they are floating and root is not attached to the substratum, so it's not even covering the function of anchorage also. And there might be, if roots are present, they might be just to absorb the mineral nutrient, and also might be help them in floating. And if they are anchoring the plant, their purpose is only to anchor the plant and not any absorption of water. So the roots are feebly developed. Even when we talk about the anatomical structure of these floating hydrophytes, in this case also you will see that the roots are poorly developed. Like lemna, which is a floating hydrophytes, contain only one root. Okay, I have told you that it contains two leaves and just one root. Wolffia doesn't contains root at all. Okay, so there is no question of root here. Then they will have root pocket, so that we have discussed. Okay, root pocket is similar to that of root cap. Roots are hairless. If roots are there, no root hairs are there. This is all absent. Okay, root hairs are absent. The epidermis, okay, when we talk about epidermis, the epidermis will be single layer. Okay, if we cut the section of a root of a free floating hydrophyte, we can see that epidermis will be there, that will be single layered. Okay, you will not find any root hairs in that. Then you will see cortex. Okay, some outer layer of cortex, in this case also one or two layers will have less intercellular spaces, whereas the other cortex will be completely modified into parenchyma. Okay, they will be arranged in that way that they will enclose a cavity. Okay, so this is the air cavity, and those parenchymatous cells are known as parenchyma if they are participating in making the cavity. Okay, then you will see a vascular cylinder. Definitely you will see a vascular cylinder. You will see it's a root, so the condition definitely will be radial and okay, exarch, and all those conditions will be there. But if you can see xylem in that, you can see phloem small amount, but the thing is that they will not be properly developed. You will see structures like this. Okay, one or two layers on the lower side. Cortex will also be having less intercellular spaces. So you can say it's an outer cortex, and it is the inner cortex. So in that the most part of the will be occupied by the cortex with air cavity. the vascular tissue will be there. You will see vascular strand is there, but differentiation between xylem and phloem will be very poor. You will see xylem, metaxylem and protoxylem like structures, but they will be not that much lignified, not very lignified. That means lignification will be very less. They will be definitely thin walled, because no hard things are required, no need of a lot of water conduction, because mostly the water is moving to every part of the plant through diffusion. So they will be poorly developed. Now this is just a TS of root of Nymphia and Eichhornia, where you can see that some outer hypodermis like layer is getting formed by the tissues which are compactly arranged. Then air spaces are there. Okay, some endodermis is also there, which is encircling the steel inside it. Okay, pith is there. Some phloem will also be definitely there. You will see xylem. Okay, but they all will be very less developed and very less differentiation in the xylem tissue, and like protoxylem and metaxylem, their differentiation will also be very less. Then in Eichhornia also you can see that how now in this you can see that the layers are this Aerenchyma formation is in slightly different way. Okay, they are forming cavities, but they are getting arranged in a different way. These Aerenchyma are packed in vertical rows and then they are encircling the cavity. Okay, so they can be arranged in different ways, but the purpose is to form air cavity or air space. Here also you will see protoxylem, metaxylem, phloem, but they all will be weakly developed. Now coming to the stem, so in this case, stem, okay, stem which is there of, or rhizome also we can call it. Sometimes suppose this in Nymphia, this is also free floating. Okay, it is attached with a long petiole, and the stem could be also in the form of rhizome. I have told you that it's a horizontal stem from where roots can also come out. So this stem or this rhizome will be well differentiated. Okay, because the stem will contain the leaves and it will actually provide them some support. So stem will be slightly differentiated when we will see the anatomy of these plant. Like in Nymphia, the stem will be well differentiated into epidermis, hypodermis, cortex, pericycle and vascular bundles. Endodermis, okay, like if we draw the structure, you will have definitely epidermis. Okay, cuticle will also be absent because still you don't require anything to prevent from desiccation. So outer some layers will be densely packed and they will be known as hypodermis. Okay, and then you will have Aerenchyma structures like this formed. So this will be cortex. Okay, then you will have endodermis. Okay, the lower layers of cortex could also be slightly packed. Then you will have endodermis. So endodermis also, a clear endodermal cells you can see. So you can see vascular bundles also. So depending on what type of plant it is, the vascular bundles will be as such. Now the cortex will contain, it will be large and possess air spaces. The cells of the cortex will be, this is endodermis, okay. The cells of the cortex will be definitely thin wall, and they will also contain chloroplast. See, many of the stems in the hydrophytes are green in color, so they perform photosynthesis. So definitely they will have chloroplast. If light is available, they will also perform photosynthesis. Now this is also important that a large number of stellate sclerids, okay, stellate sclerids also we have discussed. See, sclerid is what? It is sclerenchyma, but like fibers, it is not elongated. Fibers were elongated sclerenchyma, whereas sclerids were of like stellate. If we talk about stellate, they were star shaped sclerids. Okay, we have also discussed brachysclerid was there, that was having a different shapes. So all these sclerids are there, they have irregular shape. So particularly stellate sclerids, okay, astrosclerids, okay, this is stellate sclerids, okay. These are important feature. Presence of stellate sclerid is very important feature that is found in hydrophytes, okay. You should remember that in leaves and in stem you will find these stellate sclerids, okay. uh, and apart from this sclerid, whose function is mechanical, these all fibrous tissue, they provide support. So the sclerid will provide mechanical support, otherwise apart from them they will not have any other mechanical tissue, like no fibers will be there, no lignified trachees and vessels, very thick, all those structures will not be there. The vascular tissue will also be purely differentiated, means it won't be very easy to differentiate exactly xylem and phloem, because all those lignified tissues will also be absent. Now xylem vessels will be thin walled and phloem will be represented by narrow tubes, okay, like completely well differentiated sieve tubes and well differentiated fibers of, they all will be absent. Now this is the structure of a Gessia stem and Lymanthium stem, which shows how these is differentiated into epidermis, then hypodermis, then a lot of air spaces, in which also several layers of parenchyma in the cortex will give rise to, they will form, you can say rope, or you can say a band like structure which will enclose the air space. Then endodermis will be there, phloem will be there, definitely xylem will be there. On the outer side phloem is there, you can see lot of vascular bundles, okay, but it's a stem, so it will be conjoined vascular bundle. Phloem will be on the outer side, xylem will be on the inner side, and large pith will be there. But yes, their differentiation will not be exactly the same as we have discussed about land plants. In this particular stem also, you can see here lot of star shaped stellate sclerids are there, okay. So this is an important feature of hydrophytes. Here also you can see some compactly arranged cortex is there, outer and inner cortex, and in between you will see lot of xylem and phloem, but undifferentiated. Now coming to leaves. Now leaves are also have large air spaces in the leaf blades, because they are also floating, okay, we are talking about floating leaves. So definitely some sort of buoyancy is required for those leaves also, so that will be brought about by air spaces. Now this is very important feature that we have discussed in eichhornia, that the petiole will be swollen, okay, the petiole will be swollen. Now the swollen petiole will give rise to, will help them in buoyancy, okay, they will contain air spaces. See, when you will cut the section of this petiole, you can see lot of air spaces are there, okay. Then it will possess distinct epidermis. See, now the leaves are floating on the surface, and you would have seen like the Neelambo leaves, you would have seen. See, mostly all these leaves are green in color, they all are performing photosynthesis, they are generally flat to trap maximum sunlight, and they are also differentiated into palisade parenchyma and spongy parenchyma, because they are organized to perform photosynthesis. So they will be arranged in that way, as you have seen in those leaves which are present in the land plants. Now waxy coating will be definitely there on the upper surface to prevent water logging and to prevent the clogging of stomata with water, because stomata will be present on the upper surface, lower surface stomata will not be there. Okay. And mesophyll will be well differentiated into palisade tissue and the spongy. However, the vascular system will be poorly developed because still they do not require much conduction of water because they are surrounded by water and water can easily enter through diffusion. In this case also stellate sclerae will be present in the mesophyll tissue and their function will be to provide mechanical support. And stomata are found on the upper surface, okay, but lower surface will not have stomata. Now, these are some examples we will try to discuss. Eichhornia petiole, as I have told you that they are swollen, you can see a swollen petiole, and on the top the leaf blade is there, okay, leaf blade is there. Now, when this petiole is cut, you can see lot of small pores are there. Okay, this is the transverse cutting and this one is the longitudinal cutting. You can see a lot of small pores. These pores are actually, these are all air cavity, big air cavities are there. Now, if suppose this petiole you cut them, okay, you cut them and you immerse them in water. If you suppose if you find water hyacinth nearby, you can do this experiment by yourself. You don't require a well equipped lab, you don't require microscope also. You can take the stem, you cut them horizontally or longitudinally, try to cut them a transverse section, okay, this section you try to cut them, okay, and then you immerse it in water. Suppose this petiole is immersed in water, what you will see, air, and you press them, air bubbles will come out. This will clearly show that lot of air is trapped in the spaces which are in these air cavities, and when you will press them in water, you can see the air coming out from the in the form of bubbles. See, you can do it in air also, but you cannot observe the air bubbles. That for that you have to put them in water. So that experiment you can easily do. If suppose any water hyacinth plant is available, you can search them. If you find a pond and you try to observe that it is water hyacinth by the presence of its swollen petiole, okay, and try to do this. Now, nymphea leaf, we will try to understand this internal organization of nymphea leaf. See, it's a water lily, it's very much related to lotus plant also. If you get lotus plant, you can even try to analyze them also. You can see that lot of veins are there, okay, veins are there. So, these are vascular tissues, okay. In this vascular tissues will be slightly developed as compared to the other small plants. When you will cut the section, you will see that they will be differentiated as you find them in land plants, okay. So, when we will, so what you will see an upper epidermis and a lower epidermis, but definitely in this case, stomata will be on the upper epidermis as compared to the dicot plants in which stomata was there on the lower epidermis, okay. You will find a cuticle layer, okay. So, you will see a epidermis, okay. So, here also you can say that some stomatal pores are there. Okay, so you will see stomata. Similarly, you can draw the lower epidermis. So, you can find cuticle also on the lower upper epidermis. You will not find it on the lower epidermis. Okay, you can see a palisade layer, okay. So, in this case you will see multiple palisade layer, okay, more than 1 or 2, 3 palisade layers you can see. So definitely these palisade layer will perform, these are the mesophyll layers. Palisade parenchyma, these are all parenchymatous tissue. So they will contain chloroplast, okay. Okay, similarly. Now you will see lot of spongy parenchyma also, okay, large amount of spongy parenchyma will be there. Okay. So in that spongy parenchyma also you will see chloroplast, so they will also help in photosynthesis. But is the purpose is basically for gaseous exchange. More photosynthesis is performed by palisade parenchyma, but it's not that spongy will not. But it is not involved in gaseous exchange, so there is no stomata on the lower side. Now the important feature is presence of sclerid. Okay, so these sclerid you will see in mesophyll tissue. Okay, these are spongy parenchyma, which is in between them. You will see lot of sclerids. Okay, and vascular bundles will be there. Okay, they will be covered by a parenchymatous bundle sheath. Okay, and in that you will see definitely phloem is on the lower side. Okay, and xylem is on the upper side. Okay, but still in this the xylem and phloem will not be as much differentiated as we will see them in land plants, because still the conduction of water is not as much required because they are easily coming out through diffusion, but still it is slightly differentiated because much of the upper surface is not exposed to water, so they at least require some conduction, and phloem will be there to transport the prepared food. So all these structures will be there in a nymphia leaf. Now this also I have shown this section of a nymphia leaf has been cut. So this is the diagrammatic feature, this is the diagrammatic structure, and this is the exact, this is the, you can say it's the microscopic structure which in which the nymphia leaf was cut, and I have taken the picture. However, it's not a very good preparation, but here you can see the presence of mesophyll tissue, okay, and you can see the air cavities here, cavities, and you can see the most important is sclerids. You can see star shaped sclerids, lot of sclerids. I have kept this picture because star shaped lot of sclerids you can easily see in this picture. See here also you can see, here also you can see the vascular tissue. Okay, vascular bundle is there, fibrous tissue, some fibers, some fibers are also present in this leaf also. So you can see on that end some fibers are there, and this is the vascular tissue. Okay, um, this is the lower side, so here you will see sorry, this is the upper side, so here you will see xylem. Okay, xylem is there, then phloem will be there, and this is large air cavity. Okay, this is all air cavity. So this is the diagrammatic picture which you can see that exactly it is looking same. See, these are the pictures of the those portions of the leaf where veins are not present. So here also the thickness is slightly less, and where veins are present you will see slightly a bulging outline also at the portion where veins are there. So you can see lot of air spaces. You can see sclerids are emerging from there, coming out from there. Here also you can see lot of sclerids are, this vascular tissue is there, this is the fibrous tissue. Okay, this is the xylem, and this is some phloem portion will be there. This is the mesophyll tissue, okay, and air cavities. Now coming to amphibious plant. Now amphibious plants, as we have discussed before, they are the one which are present partly in air and partly in water. Okay, so depending on the water availability, depending on the shallowness of the water, that how much part of that plant is submerged in water. Okay, maybe 1 foot at least they might be submerged, or maybe root and some portion of the stem might be submerged, or some portion of the entire half of the plant where leaves are also there, they are also submerging. So they will adapt in that way. If the leaves are submerged leaves, so they will have adaptation of submerged plant, and if they are all growing on the upper side, they will have adaptation similar to that of land plant. So they will show both adaptation. See, abundance of mechanical tissue is there, which is adaptation of land plant, and then they will have presence of air cavity. See, even if they are growing on the in the air also, they will also contain air cavities. So this is an adaptation of hydrophyte. Now even leaves also, they will show marked differentiation of mesophyll. Presence of stomata will be there, and presence of fibrovascular bundle. See, stomata will definitely be there in the leaves which are present on the air, okay, which are exposed to the atmosphere. And this fibrous vascular bundle, okay, vascular bundles which are fibrous, which contains lignified tissues, which contains fibers, sclerenchyma. So this all will give mechanical support. This cuticle will also be present, okay, on the portions which are exposed to the land. Now see, the emergent leaves, the leaves which are on the atmosphere, okay, which is not on the ground. So in that leaves also, even in stem also, you will see presence of aerenchyma. Now the question arises that if the leaves are completely in the air, they don't need any gases exchange, they don't need any buoyancy, and mechanical tissue is there which will provide them support. See, this mechanical tissue will help the plant to be erect in the water. See, naturally, if they are floating on the water, if they are completely submerged in the water, they have the support of water. The water column is actually keeping them erect. Okay, they are helping them in supporting, and they have air cavities which is maintaining the buoyancy. But if they are above land, there is no water, and for that they require mechanical tissue. So these tissues are there. But in emergent leaves also you will find aerenchyma. So why this aerenchyma is present? When you will see the structure, where typha is a very example of a amphibious plant, you can see that large aerenchyma or continuous channels or lacuna is there, which has air filled in that. So this air is actually, this lacuna is actually connected to the under submerged portion of the plant. So this lacuna actually provides air to the submerged portion. Now this plant will not be dependent on air, is coming from the internal organs and it is flowing through the stem, through the air or gaps or lacuna, and it is coming to the under submerged portion. So there is no actually they are not dependent for the process of diffusion. So this air cavity is actually a passage through which air can move. Now these leaves are also performing photosynthesis, so they will produce oxygen. So that oxygen can be again utilized for respiration. So this oxygen and the other gases and the other oxygen more oxygen can come from the atmosphere and enter those leaves and finally to the submerged portion. Now this typha, you can see an elaborate picture, a more cellular detail of this typha. You can see here that air cavities are there. Mesophyll cells are there. You can see mesophyll cells are present on the both the sides of the part. Lower epidermis is there, upper epidermis is there. In the center you will see vascular bundles, okay, fibers will be there surrounding these vascular bundles, so they will provide more support. You will find xylem and you will find phloem, two vascular bundles you can see here. So xylem and phloem tissues, well developed xylem and phloem tissue will be there, that will be similar to that of land plant. This is also a picture of root of typha, where you can see aerenchyma is there because root will be definitely submerged in the plant, but you can see lot of mechanical tissue. Here pith is there, which is sclerenchyma, which has sclerenchyma. Now sclerenchyma has started coming. You can see xylem and phloem. Since it's a root, so radial arrangement, xylem followed by phloem patch. Okay. Then metaxylem will be there, protoxylem will be there. So all these structures will be there. Then in ranunculus stem, see this is ranunculus stem. So stem will definitely have vascular bundles in the form of ring. They will be arranged in the form of ring. So they will have a well developed vascular bundles. You can see two vascular bundles are there, but still you will see air space. Okay, this is because of forming a continuous air space for the movement of air into the submerged portion for respiration. So here you will find sclerenchymatous bundle cap, and then phloem you will see, then xylem you will see. So all these mechanical tissue will be present in these amphibious type of plants. So they will show both type of characters. Now, so with this, I come to an end with the discussion about hydrophytes. In the next lecture, we will talk about another modification or another adaptation which is there in xerophyte. And for this time, I would definitely want you all to analyze those hydrophytes, try to locate them nearby and try to analyze those hydrophytes, what morphological characters they are having, and if possible you can also try to observe the internal structures also. Okay, so thank you. We will meet again in the next lecture.